Over the last 2 decades, the treatment of human infertility has become very successful indeed. The introduction of hormones to stimulate ovulation and the introduction of assisted human conception now mean that many couples can have their own desired child in their own time. We are now promised new breakthroughs, and one special one is the introduction of recombinant follicle-stimulating hormone using DNA technology. We hope this breakthrough will lead to new advances, which we're going to see in the following program. The opportunities for recombinant technology actually started here in 1953 in the old Cavendish laboratory of the University of Cambridge. Here, 2 young scientists, Crick and Watson, made a discovery that was to change the world. They were the first to understand the structure of the DNA molecule, the very basis of life itself. And this structure has led to many fundamental advances in medicine and science. The genetic information contained in the molecule has now been utilized for advances in therapy and to make new hormones such as recombinant FSH.
The basic structure of DNA is identical for every life form on our planet. It's a double helix structure composed of a backbone with 4 nucleotides attached to it, A, G, C and T. The sequence of these nucleotides determines the genetic properties, the genes. And human DNA contains approximately 100,000 genes. FSH, or follicle-stimulating hormone, plays a key role in the development of the human ovum. It's used for controlled ovarian hyperstimulation, such as in IVF, and also for the treatment of chronic anovulation. Of all hormones, FSH is one of the most complex. It's a glycoprotein comprising 2 subunits. An alpha subunit, identical for all gonadotrophin hormones, and a beta subunit, which gives FSH its specificity. Both subunits are encoded by separate genes. Until recently, FSH could only be obtained from the urine of postmenopausal women. But now, after extensive research, Organon has managed to produce FSH by recombinant DNA technology. The first step in achieving this was identifying the as yet unknown beta subunit gene.
In the Human Genome Research Project, many thousands of hours have been devoted to the identification of single human genes. Yet so far, very few have been identified. The reason for this is simple. Searching for a single gene is like looking for a tiny needle in a very large haystack, or for the single page of an unknown book in a very large library.
But first, this library of genes had to be created. The bookcases in this library are dishes with E. coli bacteria, each of which contains a fragment of the human DNA. Next, the search for the beta subunit gene had to be narrowed down to, let's say, 1 book, or 1 specific DNA fragment, which might still contain many genes. This was done by adding synthesized DNA fragments called probes. These probes were specifically designed using the knowledge of the beta subunit protein sequence. When the probe matched an FSH beta gene fragment, this showed up as a dark spot. So now the book was identified and the search for the beta gene page could begin. For this, the book first had to be divided into chapters. So the DNA fragment was split into smaller pieces with the help of highly specific DNA cutting enzymes. These pieces were separated by gel electrophoresis. Visualized under ultraviolet light, the larger DNA pieces were seen as bands at the top of the gel. And one of these bands had to hide the beta gene page. To actually read this page and confirm the presence of the gene, the sequence of the A, G, C and T nucleotides had to be obtained. So thousands of sequences were digitized and further analyzed by computers. Newly found sequences had to be compared with the information deduced from the FSH protein. So, at a certain moment, a positive match had to show up.
Several T's and these sequences here and here, I think we have some positive clones now. You see here.
Now the Organon research team had found both FSH subunit genes, and the hormone was finally within reach. This three-dimensional projection shows the FSH molecule. The alpha subunit is green and the beta subunit is blue. But subunits alone don't make hormones. Biologically active FSH also contains carbohydrate side chains, here shown in purple. In red, the part of the molecule that binds to the FSH receptor. An advanced cell line had to be selected to produce such a complex system. After careful selection, the Chinese hamster ovary cell line was chosen. These cells were transfected with the FSH genes. The human FSH genes were accepted into the nucleus of the cell as if they were its own. After introduction of the FSH genes into the chromosomes of the host cell, it's now capable of producing biologically active FSH. The FSH alpha and beta subunits are synthesized and assembled. Simultaneously, carbohydrate side chains are added in a complex sequence of biochemical reactions. This essential process is called glycosylation, and the specific way it's done gives Organon's recombinant FSH its unique profile. The next stage was to develop a reliable production process. So, well-defined and tightly controlled conditions had to be established for optimal growth of the cells and FSH production. The last stage was purification of the hormone from the cell culture supernatant. This yielded a more than 99% pure hormone. And so only one name seemed to be appropriate for this new product: Puregon.
And there it was, recombinant FSH. It had taken 10 and more years to prepare. Splendid, but would it work? It was time to give it to the endocrinology laboratory for tests in animals before the clinical trials began.
Every success brought a new challenge. Now the players of the endocrinology lab had to join in to test the new product for efficacy and safety. The biological activity of Puregon was tested using cell lines and animal models. Results of these studies revealed that Puregon was at least as potent as urinary FSH, though pharmacokinetic studies in animals showed that Puregon remained in the blood for a shorter duration. This might be due to the small differences between the carbohydrate side chains in Puregon and urinary FSH. Beside these remarkable observations, an extensive toxicological program also confirmed the safety of the compound. In preparation for clinical trials, a master cell bank was created and stored. This master cell bank is the basis for each and every batch of Puregon and ensures batch-to-batch consistency. Every batch is produced in a fermenter where the cells, all clones from the master cell bank, are grown for several months. A new pharmaceutical formula was developed using lyospheres, freeze-dried droplets of the FSH compound.
So far, so good. In the endocrinology laboratory, Puregon had proved to be a most interesting compound. Now it was time for clinical trials, because there were so many questions to be answered. Would Puregon induce the ovulation of sufficient oocytes? And how about fertilization rates? Would they be acceptable? And what was the chance of side effects in the patients? These were exciting times for Organon and for the scientists and clinicians who had now joined in the research.
The first study was performed by Dr. Fausser's group at Dijkzicht Hospital in Rotterdam, the Netherlands. Its objective was to test the biological effects of Puregon on volunteers who had no FSH production of their own. Puregon appeared to be safe and induced development of follicles. Here, finally, was clear evidence of Puregon's effectiveness. It was also concluded that follicular development could even be achieved in the absence of LH. Another study was undertaken by the team of Professor De Vrooy and Professor van Sterklichen at the Academic Hospital in Brussels. This is Europe's largest IVF clinic. Here, the now famous ICSI technique to treat male infertility was developed. A more extensive IVF study in 50 patients was performed. Various GnRH agonists were used to reduce endogenous FSH and LH production. Once again, the outcome was highly satisfactory. All patients developed sufficient and mature oocytes. Even the very low LH levels were sufficient to support adequate estradiol production. And a milestone was reached, the first baby, born as a direct result of treatment with Puregon. Large-scale studies were then carried out in 14 countries, including the Diakonessen Hospital in Voorburg, the Netherlands. Most of these compared the effectiveness and safety of Puregon with urinary FSH. The most important clinical trial was the largest randomized study ever performed in IVF. More than 1,000 patients in 18 centers were involved. With so large a study, it was possible to assess reliably the true efficacy and safety of Puregon. It proved unambiguously that after Puregon treatment, more follicles developed, more oocytes were retrieved, and more embryos were obtained compared to urinary FSH. But the fertility specialists also discovered another benefit. Patients needed a lower total dose and a shorter treatment period with Puregon. There was 1 chance in 5 of becoming pregnant after treatment with urinary FSH, but 1 chance in 4 after Puregon treatment. A highly significant difference. In other words, a 25% higher chance of a baby with Puregon. The conclusion is inevitable. Compared to urinary FSH, treatment with Puregon results in more follicles, more oocytes, and more pregnancies, using a lower total dose during a shorter treatment period. But that's not all. Due to its high purity, Puregon can safely be given subcutaneously, more convenient for patients. One further conclusion of the clinical trials was that Puregon's increased effectiveness called for new formulations, 50, 100 and 150 unit ampules for better fine-tuning.
Puregon is a remarkable medicine. It offers better treatments for infertility. Patients will benefit because their treatment will be more convenient and their chances of pregnancy will be greater. And lastly, and most important, it offers more happy families for many more infertile couples.